Okay, so let's finish the left slides of these lecture 3, then we move on to lecture 4. Lecture 4 has a lot of material. Now we're going to get on to the structure of the bacteria. So we're using bacteria as an example, so ribosome. First of all, we will talk about the detail in the examination 3. Here is just some introduction. Ribosome is the place to synthesize protein. And basically there is two different types of the ribosome. Some of the ribosome is on the cell membrane system. Some of the ribosome is inside of the cell. So there is two different types. We don't want to go to the detail of that. We just give you the simple knowledge behind it. Okay, ribosome. If we talk about a bacteria, it is 70S. It has two subunits. A subunit is relatively small, another is a big chunk one. And the big one is 50S, and the small one is 30S. Now here comes the very tricky stuff. This is add up is 70S. It is not 80S. Why? That's a very simple question for you. This S, the Berg unit. This is not molecular weight. So don't simply add it. It's not going to add up. What is the Berg units? Which is the parameter to testing the precipitation. And how we do the precipitation, let's say you had a tube. Okay, I'm going to put the isolated protein from the ribosome, and then I do the centrifuge. After centrifuge, it will be spin down. And how fast it's going to spin down, then you have a unit for that. It's called the Berg unit. So it is not a mass molecular weight or some simple number you add up. It's not going to add up. The whole Berg units, you can say the rate of the spin down, the precipitation of the whole ribosome is 70S, and composed by two subunits, which is 50S and 30S. Now, what is inside the 50S? Inside the 50S, you have 23S subunits and 5S subunits. This is later on, you know, they will be help for this large subunit to cover with attach with the small subunits during the RNA translation, which means synthesize protein. How about here the 30S subunits? There is a 16S rRNA, which means there is another subunit there. And this is 16S rRNA is very important. Number one, they have a very conservative sequence from V6 to V8. It's a universal conservative sequence for bacteria. Therefore, we come out with so-called 16S rRNA biotechnology. If you are doing some of the research with faculty in medical school at WVU, I believe you heard about that before. The second thing is that you know during the protein synthesis and RNA translation, the 16 RNA used to recognize the so-called GAA, AGG and AGG. It is a small conservative sequence for the Shine-Diagonal. We call it the Shine-Diagonal sequence. That one is for initiate the RNA translation. Okay, so I just want to let you know, this 16S RNA underneath the 30S subunits is very important. Now, obviously, the eukaryotes, which is the fungi, the ribosome, which is ADS. So we don't talk about too much here because we don't talk about fungi. Nucleids. Nucleids will be very simple to talk about here because in the examination 3 we will go detail DNA structure and then followed by the central dogma, the DNA replication, transcription, and the translation. So if you remember, this is we call it the central dogma, which is we call DNA replication. And DNA will do the transcription, become RNA, and then RNA will do the translation, become protein. Here, we just very simple to talk about the DNA. It is a circulated, double-stranded, you know that. And usually it looks like this. If you remember in your other classes, it's called semi-conservative replication. It's a double helix, and we call it negative supercoiled double helix. And you know that it's composed by gene base A, G, T, C, and A complementary with T, and G complementary with C, which is right there. And so it's very simple, and the nucleic gas is usually the location of is in the chromosome. Now, specifically for bacteria, there has come out a very special DNA, which means it is around 30 base pair. Most of the time is circulated, or it is linear, could be linear, a single linear DNA sequence. This very small size DNA sequence, we give them a name called a plasmid. And this plasmid, they could do, which is their individual replication by itself. They also could do, if you have a large chromosome, and in the chromosome, this guy can integrate it and then become part of the whole chromosome gene sequence. And this is, we call it an episome. Now, the plasmid is very important. Sometimes they have a very special function. When we talk about antibiotic resistant, lots of the time it is started from the plasmid. The sequence has been changing. Make sure they make it become antibiotic resistant. So we call it all plasmid. And this all plasmid, if they go to the environment brought by the bacteria, they could be naturally sometimes taken by the other bacteria, then become one of the formation of the genetic communication. We call it a transformation. That could be just nakedly taken by another bacteria. That's called transformation. Okay, so this is, we gave you a very simple introduction about the plasmid. Some of the external structure of the bacteria we will talk about is phyla, fimbrial, and talk about the flagella a little bit more. Here is something pretty straightforward. Fimbrial and phyla, or phyla, which is short, thin, hair-like appendages. They say they have some of the motility function, but I will not pay much attention to that. It's barely, maybe tumbling a little bit, but it's not really moving function compared to the flagella. Major function for that is DNA uptake. This comes up another communication for the genetic information, which is called conjugation. The conjugation, if you very simply to say, I have this chromosome right here. It's a bacteria. This is another bacteria right here, let's say. It's a chromosome. and then I find it's a breach. The breach is right here. This is usually is a filling. Some of the bacteria we call it sex filling. And you can say it's a joke, it's like a sexual intercourse with the bacteria, and they communicate with the information. Okay, from here to here. So that's the conjugation, another way for the communication of the bacteria. So that's a function for the filling. We just talked very briefly. Now we move on to the flagella. Flagella, we talked about already. We can use flagella stain to testing and to find the morphology and the presence absence of the flagella. Now, first of all, flagella is composed by a protein called flagellin. Second, the location of the flagella, making the bacteria into 4 different categories. Sometimes a bacteria only have 1 flagella at the end. Therefore, that bacteria have a very limited moving ability. Let's say the slides right here is Pseudomonas. They only do a very limited moving. We call it the monotrichos. Yes, okay. Then we have another example. Is a bacteria will have 2 flagella at each end. This is called amphitrichos. When you have a 2 flagella at each end, the moving capability started to increase a little bit. Then you have another situation, which means lots of the flagella will be stacked or accumulated at one end. This is called the lophotrichos. I want to make sure the spelling. L-O-P-H, lophotrichos. Now the last one, you will see in the lab, we will mention this bacteria very often. The flagella has been covered the whole surface of the bacterial body. And right here you see the slides. P is Proteus vulgarius. It's covered by everything. It's spread everywhere. It has a very strong moving capability. This is called petritrichos. Petritrichos. This one is Proteus vulgarius. Is a typical bacteria. The soil pathogen has this structure, a very strong moving capability. Now, how strong it is? We will mention lots of the bacteria have single colony. This bacteria will not forming a very easy isolated single colony. It will be like this, like a flower. So we call it swamp. Okay? Now, regarding the flagella, I also want to briefly mention there is one parasite we can say it's have been involved in the female sexual transmitted disease is Trichomonas vaginalis. They cause like itching, abnormal discharge from the vaginal tract, from the vaginal tract. What it looks like, because it's eukaryote, so their flagella is very interesting. It looks like this. Sorry, 1, 2, 3, 4, 5, 6, 7, 8. There is one more there, so it's 9. And at the middle, they have 2. We call it a 9 plus 2. This is typically for eukaryotes, which is a fungi. You think a fungi, the flagella looks like that. Okay, also composed by flagella. So that's more complicated. And then, because of the 9 plus 2 structure, the moving capability is very strong. We just briefly show you an example for endoflagella. Treponema palladium. Remember this bacteria we talk about. It's a very special bacteria. We call it spiral sheets. Because it's very soft, so you cannot use a general simple stem or gram stem to do the observation of the morphology. Therefore, we were using dark field microscope. This is exactly what it looks like. And then I said it's very special structure for their movement. It's also a flagella, but we don't call it a flagella. It's an endoflagella. It will be forming corkscrew-like movement. Okay, look at that. Because the bacteria has flagella, they can do a real moving. If any of the signal comes, regardless it's a bad or good one, you can put a candy there, they're going to go there. If there is some stress environment, their cell membrane system can do the censoring, then they're going to move away. So they go there and move away like you take a round trip from here to Pittsburgh International Airport. So we call a special terminology, is called chemotaxis. That is movement of cells toward chemical attractors or away from chemical repels. Make sure you understand it's a round trip. Sometimes they can move without flagella, but most of the time they are moving with flagella. So you can see there is a good example here. Okay? Last slides, endospore. We want to talk about this a little bit detail. Luckily, you're going to do the endospore stem real quick, so it's a good time we discuss about this. If you're lab 1, you can just think about this like a review because you already did. And we mentioned briefly about that, but if you're lab 2, 3, 4, it's a good time we're really going to catch the knowledge of the endospore. So bacteria endospore. The first thing first, we mentioned about there is 2 major bacteria has endospore. Is Clostridium and the Bacillus. What are the Clostridium we will talk about in this class later on? That is Clostridium tetanus. Clostridium botulinum. Botulinum. Clostridium difficile. And Clostridium perfringens. We will talk very detailed about all of them, the characteristics, the disease associated with that in the examination too. But I tell you one thing, this is a biosafety level 3 pathogen. So we cannot do in the lab. Our lab is certified, is only biosafety 1 and 2. The university I remember can do this is in University of Minnesota at Twin City. Their food science department have some special lab to do the Clostridium. It is a big issue for the food industry. Remember, Clostridium difficile will cause colitis of the intestinal area, transport from the fecal sample. Long time ago, we think it's only for the human being, but right now, it's an emerging pathogen for the food supply chain. Clostridium botulinum, you know about that, is low acid food, the canned food. Remember, we talk about in West Virginia, the requirements is a pH value less than 4.0, because if you're above that, it's a very nice environment for the endospore to form by Clostridium botulism. Clostridium tetanus will cause lockjaw. If there is a heavy metal contaminated, then you cut your skin. You do not do the sanitizing and do any treatment immediately. Your whole muscle may be like become a tough muscle. That's called a lockjaw. Okay. Clostridium perfringens, they have a capability to separate the muscle. They break down the sugar between the muscle and generate a gas. We call it a gas gangrene. And we will talk about these details. Bacillus, Bacillus cereus, and the Bacillus subtilis. these two things usually happen is in a rice. If you have a bacillus, you have a rice left from the dining center, and then you dorm for about 10 days. Very easy, the bacillus spore will have there, so throw them away. Okay, so this is Clostridium bacillus, is a major bacteria will have endospore. Now, what is the endospore? Endospore is a protection structure. What means a protection structure? Which means they do not have reproductive function. That means if the mother cell has endospore, does not mean the daughter cell will automatically has endospores. The reason is it is only in a stress environment will forming endospore. Then we have another question. Why endospore can survive in a stress environment? Because it's a very special structure. We talked about this already. At the beginning, you will see there is a coat. Then we talk about there is a cortex. And the cortex, the major components is the polysaccharides. And it's a very similar structure, like peptide glycan. Then you go there, you have a cell wall, cell membrane system, very similar to that. But this is not a big deal. The big deal is right in the middle. Right in the middle, there is two major special structures. Number one, calcium diproconic acids, calcium DPA. The second one is a small acid soluble protein. Because of this special structure, the endospore will be forming. Now, once we talk about the endospore, we talk about the location is important because the location is genetic dependent. And there is three possible locations we already mentioned. It could be in the middle. That's why we call it a center. It could be at the end, like you go to the airport. We call it a terminal. And most of the time, especially for the bacillus, they will be right in the center, like a bubble. We call it a sub-terminal. And it is genetic dependent. We know the location, we know the symptoms, we get isolation from the patient, we can guess after doing the endospore scan what it looks like. And because the endospore is a very rigid structure, so we using malachite green and using heat steaming, we can do the endospore stain. That's what we're going to test. Now, I want to tell you one thing in the real life, what's going to be happening in endospore. You have to avoid doing this. This is a true story. One of my friends in University of Louisville, not too far away, okay, he's a faculty. This is what happened. The day in the morning, that's when he told me the story. He's going to prepare something for his family. What they have? A beef stew. Okay? A tomato. Now, of course, we're going to put water. Some piece of carrots. Like there are carrots. Okay, then they make a soup. The soup, what do they do? Is like cooked at 55 degrees Celsius, about 8 hours. So in the morning, the guy cooked the soup, said, told his wife, said, let's go hiking, and 2 kids. The 2 dogs with. The soup has beef stew, beef trimming, tomato, and some carrots. Something else, maybe some lemonade, I forgot he talked about that. Once you cook him, beef stew, what they're going to generate? We will talk real quick. beef extract. Once you see the beef extract, it's a brownish color. It's a big, nice environment for bacterial growth. It is a bacterial media, I tell you. Kind of like you're making a bacterial media. Where the carrots come from? Where the tomatoes come from? Yeah, of course they come from supermarkets. Where the original come from? Come from soil, is that right? They all coming from soil. Oh, I remember he put some gingers in. That's also coming from soil. Where these endospore bacteria really come from? You think Clostridium bacillus, where the originally come from? Come from the soil. These are contaminated, then you trim it. They're still there. The key issue here is that you are cooking at 55 degrees Celsius for 8 hours. You try to make a soup, but make sure, remember, this is a low temperature. When this low temperature heated, lots of the normal bacteria, the background flora, or we say, will be killed. This is, we call it a mild heat. This mild heat will kill lots of the background bacteria. And it creates a stress environment for endospore. So what happened is, you know, the bacteria in the real life, they actually have a competition with each other. Sometimes you have an E. coli grow there. Maybe Bacillus or Clostridium will be inhibited a little bit. Now you use a mild heat, they all died. And you create a stress environment, let the endospore bacteria to survive, to grow, and multiplication. So what happened later on? The whole pot is dominated with those endospores, and you won't see it. So you have 8-hour hiking. I believe they go to the Mammoth Cave, sometimes which is in the south of the 2 hours away from Louisville. They go home at 5 o'clock in the evening, and the husband said, okay, let's have a soup. Everybody have a soup. The dog have a soup. 6 hours later, they all have a vomiting, diarrhea, nausea, all those things. And they go to Louisville Medical Center and tested with Clostridium, botulinum, another species I forgot, the dew bacterial culture endospore is in their stool sample and the vomited sample. So that's why I tell you one thing. You should be avoid is this type of the cooking. This we will mention later on. The temperature is very important for the bacterial growth. You should avoid 40 to 140 degrees Fahrenheit. This is a dangerous temperature zone. You can do the calculation. That is 48 minus 32 multiplied by 5 divided by 9. Can you do that? It's 40 by 9. What's that? Around 4.4 degrees Celsius. Avoid that. What is 140? Around 60 degrees Celsius. 55 is well in this dangerous zone. That's a favorable environment and temperature for the bacterial growth, especially for the endospore. So you should avoid is low temperature, long time cooking. Then make the beef really good, because the beef, we talk about the flavor, juiciness, and the tenderness. It's really tender. However, you improve the quality of your soup, but you forget about the safety of your soup. So we have to do the balance with the quality and the safety. So that's a good example to show you the endospore. Now the endospore have another tricky thing is that it could be there forever. It could be there for a week, for months, for a year, or 10 years. There is some older culture can be there for 1,000 years, the endospore. They always in a dormant status. But sometimes when the condition changes, let's say these stress environments are removed, they will become a normal bacteria. We call it germination. And generate endospore, we call it sporation. So we have these two terminologies to explain it. It could be the sporation status, the dormant status for the rest of their life, or it could become a normal bacteria, which is we call it a vegetative cell. That's called germination. So it's very tricky lots of the time. Okay? So this is what we finished. Talk about the examination 1, slide 3, and the lecture 4. Now we're going to move on to the last one, which is talk about lecture 4 is microbial growth and nutrition. So I'll just temporarily stop this video.